Okay, very good morning, my dear friends. In the regarding of CPGET 2023 PG entrance online coaching, free online coaching for all competitive exams in plant sciences, eminent personalities are delivering their lectures. Already previously, plant taxonomy has completed. Plant taxonomy one class has completed, my dear friends. So now we would like to invite one of the eminent faculty in plant sciences, specialization plant genetics or genetics and molecular biology. So Sir is going to explain about the plant genetics class. Today's our eminent resource person, Dr. Bhageerath Garu, Assistant Professor, Department of Botany, Government Degree College, Rangasaipet, Warangal District. Sir, please welcome. And I will giving you a sharing option, sir. Please share your screen and start your session, sir.
Thank you. Thank you very much, sir. I am very happy to associate in this platform to coaching for CPGET Botany PG entrance examination. So the slide is visible.
No, sir. It is. Please click on that PDF format once again. Slide is not visible.
Now it is visible.
No, sir. No.
No.
Please click on that linkage.
Now itself it is visible.
It is visible. It is visible, but PDF is not enlarged, sir.
Enlarged. Okay, it's visible.
PDF is not open still now.
Still not open. Okay, okay. I will once again share. Okay, very nice.
Yeah. Please you proceed.
Okay. So in the yesterday class, we discuss non-Mendelian ratios. How the Mendel dihybrid ratio 9:3:3:1 modified in different cases, in different experiments. After Mendel, many geneticists are conducted experiments on plants and animals. They revealed that the Mendel laws are wrong when a chromosome containing many genes, in between the genes, some interaction is going on. That interactions affect the traits or characteristics of the organisms. That's why the gene action, which says that the non-Mendelian ratios are seen in different values, that is 12:3:1 or 9:3:4 or 9:7. These are the different ratios we are yesterday discussed, and we can explain in a needful manner. So today we will go for another topic in the genetics, that is linkage and crossing. Linkage and crossing. So just we can go for recapitulate the discovery of linkage. So in 1900, the Mendel's work was rediscovered, and scientists were testing his theories with as many different genes and organisms as possible. That's why Carl Correns, Victor Hugo de Vries, and Erwin Schermark, these three scientists are separately rediscovered Mendelian principles in 1900. Later on, William Bateson and R.C. Punnett were working with several traits in sweet pea plant. So Mendel conducted experiments on garden pea plant, but William Bateson and R.C. Punnett working with several traits in sweet pea. The scientific name is Lathyrus odoratus. So this William Bateson and R.C. Punnett, they proposed another formation, another concept, that is linkage concept. Linkage is nothing but any chromosome which containing many genes. So if gene number is exceed when compared to the chromosome number in any living organism or plant. So that just you can observe what is meant by linkage. So it is a phenomenon of inheritance of genes together and retain the parental characters even in offsprings also. Just you can observe the tendency of genes. Tendency means oka pravrutti, oka nature annamata. The tendency of genes to remain together in the original combination during inheritance is called as linkage. Linkage phenomena, before going to the linkage phenomena, all genes are arranged in a linear fashion in the chromosome. So first time, the phenomenon of linkage was firstly reported by Bateson and Punnett in 1906. So later on, T.H. Morgan put forward a theory of linkage and conclude the two aspects of the linkage. The two aspects of the linkage is coupling and repulsion, were the two phases of single phenomenon of linkage. The linkage phenomenon is explained by two aspects, that is coupling and repulsion. So before going to the illustration, so what is meant by linkage? Linkage is defined genetically as the failure of two genes to assert independently. In the Mendel dihybrid ratio, we can explain about one law. What is that law? The law of independent of assertment of independent. So the law of independent assertment of genes, which is failure due to the one phenomena. What is that phenomena? What is that phenomena? Linkage. So linkage is nothing but it is the tendency of the genes, the togetherness of genes. So all the genes are present on the same chromosome, which are linked, and they transmitted to the parent or the same gamete, and which is enter into the offsprings also. So linkage occur when two genes are close to each other on the same chromosome. But in the case of Mendel, all the 7 pairs of the character responsible genes are non-linked. There is no linkage phenomena in the Mendelian genetics or Mendelian principles. Why? Because all the 7 pairs of the genes are present on different chromosomes. That's why there is no linkage in the Mendelian principles. So just one thing you can observe, the strength of the linkage depends upon the distance of the genes. So for example, this chromosome containing 3 genes, A, B, and C, and D, like this. So the distance between the two genes, it shows that strength of the linkage. The strength of the linkage is inversely proportional to distance between the genes. This is one of the formula you can notice. So the distance between the genes. So if the distance between the two genes very smaller, that shows what strong linkage is there. Are you followed? Hello? Please respond to my points.
Yes.
The distance between the two genes is very smaller. That shows what a strong linkage is there. So in the same way, A and D, the distance is far away when compared to the A-B genes. So that's why it shows less strength in between AD genes. So you can observe that the linkage, the strength of the linkage is inversely proportional to distance between the genes. So linkage is based on the between the two genes. It explain about frequency of crossing over also. So just you can go for next slide. So crossing over occurs in prophase of meiosis 1. Everybody knows very well, where homologous chromosomes break at identical to union, break and reunion with each other. So types of linkage. So before going to the types of linkage, how the linkage phenomena is detected. So just we can go for the one example. Okay. Detecting the linkage through the test cross. Detecting the linkage through the test cross. So the test cross result in the dihybrid test cross results. So what is the dihybrid test cross results explaining about that? So the ratio is, just you can, what is the ratio of dihybrid test cross?
1:1:1:1:1.
Yes, 4 1s, it indicates that. But when a linkage phenomena observed in the genes, we not get this type of ratio. Instead of 4 1s, we can get 7:1:1:7. This indicates linkage phenomena is detected in the genes. So this type of linkage phenomena was observed in Lathyrus odoratus by taking two characters, that is the flower color and the pollen shape. So these two characteristics reveals that the 7:1:1:7. So for example, so if I will go through the chart, just you can go for here, is there. See this. So flower color purple and red, pollen shape long and round. So you can go through the test cross, the FN individual cross with recessive parent. So, but in the normal dihybrid ratio, so this is the true parent plants, which are homozygous condition, purple and long and red round. So purple long pollen crossed with red round pollen, we are getting F1 plants all heterozygous, and F1 self-pollination, we are getting this ratio. So as per Mendel ratio, we are getting what is that? 9:3:3:1. But in the experimental, we are not getting this ratio 9:3:3:1. So instead of that, we are getting a different ratio. If 284, 21, 21, 55, these values indicates 9:3:3:1. But experimentally, we got these values, 215, 71, and 71, 24. It is shows that a wrong Mendelian. It is not applicable independent of assortment. So just nothing but the P and L, both the genes are linked genes. Both the genes are linked genes. Are followed? So that's why we are getting a different ratio. So F2 progeny do not appear in the ratio 9:3:3:1 ratio expected with independent assortment. There is no independent of assortment in these genes. So this experiment reveals that the linkage phenomena observed in this example. So for example, in the test cross 7:1:1:7, it indicates that coupling phase. And sometimes we are getting this type of ratio also. What is that ratio? 1:7:7:1 also. So this ratio indicates repulsion phase. So are you followed? T.H. Morgan put forth the theory of linkage, conclude the coupling and repulsion were two phases of single phenomena of linkage. So this good example explains about the linkage phenomena. Are you followed?
Yes, sir.
Okay. Next, we can go for that.
Yes,
Types of linkage. Okay, now we take types of linkage. Types of linkage, two types of linkages are there. One is complete linkage, another one is incomplete or partial linkage. Complete linkage means, so for example, A, B, B, this is one parental plant, cross with recessive, small a, small b, and small b. So we are getting F1, AA, and BB. You know very well. From this, we are getting 4 types of gametes. So here, if complete linkage is there means there is no non-parental combinations. In the offsprings, we never see non-parental combinations. That means total offsprings are similar to the parental characters. So that is indicates that complete linkage, 100% linkage is present. So example, Drosophila melanogaster. So I will show one example here. So the complete linkage, the phenomenon of inheritance of completely linked genes is called complete linkage. In complete linkage, the linked genes which are closely located on the chromosome do not separate and inheritance togetherly. Example, X chromosome of Drosophila males shows complete linkage. I will show here. Example is there. Where is that? Is there? No. That complete linkage is nothing but all the characters which present in the offsprings are similar to the parents. For example, what are the 2 characters in the parents? We can see same characters in the offsprings. That is indicate complete linkage 100%. The A and B, 2 genes are present. A and B genes are present. That is, this is the homologous chromosome, and B gene is here, and the respective another homologous chromosome. This is called as this. In between these 2 genes, linkage is there. They never separated until and unless crossing over takes place. No crossing over is there. Definitely, we are getting only 2 types of gametes. AB gamete and another one is small AB gamete. Are you followed? So these 2 gametes are formed, and by the union of these 2 gametes, we are getting only parental characters. No non-parental combinations in the complete linkage phenomena. Is it clear? Okay. Next, incomplete linkage. Incomplete linkage means the phenomenon of inheritance of incomplete linked genes is so incomplete.
Hey, hero.
The linked genes which are distantly located on the same chromosome and have a chance to separate by crossing over are known as incompletely linked. So for that is nothing but what we can explain here.
Computer lab. Third room number 3, computer lab.
This is the chromosome A and B and C. So here another one is A.
మంచిగా ఒకటే తీసుకపోర్రి రెండు కాదు అయ్యో ఎట్లా బతుకుతారమ్మా మీరు గీది లావటకపోతే ఎట్లా బతుకుతారు బాబు చూడుగా ఆయన మీరు ఉట్టక ముందు 30 ఏళ్ళు 35 ఏళ్ళ నుంచి పని చేస్తుండే ఆయన
సో A B but we are getting
okay, no problem. అలా పెట్టు ఆ పిల్లోడు లోపల పెడతా
A B capital C.
ఏ మావని ఇక స్కూల్లో టీసీకి అప్లికేషన్ పెట్టవేనా స్కూల్ కు టీసీ అండ్ ఏమైనా ఖాళీ ఏడు ఉంటామన్నా మనం పనులు
తిరుపతి సార్ మ్యూట్ యువర్
ఆన్లైన్ క్లాసు అట అది
తిరుపతి సార్
ఒకసారి రద్దు బాబు నీకు కష్టమైతే
తిరుపతి సార్ మ్యూట్ యువర్ సెల్ఫ్
అక్కడ పెడతా పిల్లోడు లోపల పెడతాడు లోపల ఒకదాని మీద ఒకటి పెట్టిద్దాం అమ్ములకు పిల్లలు వచ్చినాక నింపిద్దాం
సార్
ఇక్కడ పెట్టు ఆయన ఉండవుతాడు ఆ పిల్లోనికి
సార్ మ్యూట్ యువర్ సెల్ఫ్ సార్ just you can go through the incomplete linkage means the linked genes are separated. The linked genes are separated by the one of the phenomenon that is known as crossing over. So at a single point, the 2 chromatids are exchanged.
So it will return a single value. It will return a single value to the caller function.
the two chromatids are exchanged together, break the two chromatids and reunion takes place. That's why the AC, the linkage is breakdown. We are C here, capital C, but after crossing over, you can see in the place of capital C, what is that? Small c gene is present. So that's why a crossing over takes place. We are getting non-parental combination, non-parental combination. So in generally, we can see in the test cross, 7:1:1:7. The 7 plus 7 is similar to the parental characters. 1 plus 1 only is the non-parental character. These non-parental characters are formed by the exchange or replacement of the genes of homologous chromosomes due to the crossing over. And whenever crossing over takes place in a homologous chromosome, the gene position is altered. When gene position is altered, the linkage is break. The linkage, the togetherness of the AC gene breakdown, and the capital C place replaced by small c. That's why we are getting this 1 plus 1 non-parental combination. So in the incomplete linkage, the linked genes which are distantly located, if the distance is very smaller, there is no chance of separation. The distance is far away, there is a chance to separate the linked genes by crossing over. This is known as incomplete linkage. Are you followed? Are you followed my lecture?
Yes, sir.
Okay, sir. Yes.
So in GMAs, color and shape of the grain show incomplete linkage. In Drosophila melanogaster, X chromosome of Drosophila melanogaster, it shows complete linkage. So these are the best examples for complete linkage and incomplete linkage. So these are the, if some frequency of crossing over also occurs between the linked genes, it is known as incomplete or partial linkage. Example, maize plant, pea plant, Drosophila female, and several other organisms also. Complete linkage present in males of Drosophila, females of silk. These are the two examples for complete linkage and incomplete linkage. So once we can go for next part of the, based on the crossing over, we can explain like, so what is meant by coupling phase? What is meant by repulsion phase? The T.H. Morgan in 1911, he explained the theory of linkage by two aspects, that is coupling phase and repulsion phase. Coupling phase means the two dominant alleles which are brought to the gamete from a single parent, are the two recessive allelic genes which are brought to the gamete from a single parent, that is known as coupling phase. Coupling phase లో ఏం జరుగుతుందంటే టూ డామినెంట్ జీన్స్ అనేటివి ఒక పేరెంట్ నుంచే తీసుకురాబడతాయి గ్యామెట్ లోకి. So the two dominant alleles present on the same chromosome and recessive alleles also present on the same chromosome shows coupling phase. So coupling phase is also, we can say, cis phase అంటారు దీన్ని. మనం ఏమనొచ్చు? Cis phase. Cis phase means, so you can chemistry, you studied the cis-butane, trans-butane. Is there? No. So this is the chromosome. So capital A and capital B. So both the dominant genes coming from the single parent. So both the recessive alleles coming from the single parent, that shows coupling phase. But sometimes what happened, repulsion phase is also occurs. Repulsion phase means one dominant gene and one recessive gene coming from the parent, or two dominant genes which brought to the gametes from two different parents. So here what we are explaining here, capital A, small a is there, but here capital B is there. This is called as transposition. What is that? Transposition. దీన్నేమో cis position అంటాం, దీన్నేమంటాం transposition అంటాం. So transposition and cis positions which explaining about the coupling phase and as well as repulsion phase. So just you can go through this one. This is the trans. Trans ante the two dominant genes brought to the gametes by different parents. Here what? The two dominant genes brought to the gametes by single parent. Single parent నుంచి వస్తే two dominant genes, దాన్ని ఏమంటాం మనం coupling phase అని చెప్తాం. Two dominant genes coming to the gametes by different parent plants, that indicates repulsion phase. See here, dominant alleles of the some genes are linked with recessive alleles. ఇక్కడ ఎవరెవరి మధ్య లింకేజ్ ఫినామినా ఉంది? డామినెంట్ జీన్ కి రెసెసివ్ జీన్ కి మధ్య లింకేజ్ ఫినామినా ఉంది. ఇక్కడ టూ డామినెంట్ జీన్స్ మధ్య లింకేజ్ ఫినామినా ఉంది. The linkage phenomena observed in between the two dominant genes that indicate coupling phase. A dominant and a recessive genes are linked on the same chromosome, that shows repulsion phase. Are you followed?
Yes, sir.
So next, so repulsion phase is also completed. The key difference between coupling and repulsion is that coupling refers to the linkage of two dominant or recessive alleles, while repulsion refers to the linkage of dominant alleles with recessive alleles. So ikkada linkage anedi rendu dominant genes madhya unda, rendu recessive alleles madhya unda anede gurtupeట్టుకోండి. Oka dominant gene oka recessive madhya ganaka unte, that is called as repulsion. So types of linkage, we can explain that this is a arrangement of the genes on the chromosome. It indicates that coupling conformation, and this is called as a repulsion conformation. So one dominant allele is linked with recessive allele of the second gene. This type of gene arrangement is called what? What is that? Trans arrangement. What is that? Trans arrangement. This is called what arrangement? Cis arrangement. Cis butane, N-butane చదువుతారు కదా మీరు. 1-chlorobutane, 4-chlorobutane. You studied in the chemistry. That is called geometrical isomerism lo chavutaru. So in the same way, cis and trans arrangement of the genes, which explaining about the linkage phenomena. So difference between the coupling and repulsion. Coupling refers to the linkage of two dominant genes or two recessive alleles. Repulsion refers linkage dominant allele with recessive allele. Enter the gamete. Enter the same gamete. Both the genes, A and B, both the genes are enter into one gamete. But here, enter into different gamete, because A with small b, B with small a. So that's why enter into the different gametes. Genes come from the same parent, genes come from the different parents. Inheritance, both are inherited together. Ante ikkada both the dominant genes are together, but here inherited separately. Cis arrangement, trans arrangement. So we are getting what ratio if it is in the cis arrangement? 7:1:1:7. It is in transposition. What we are obtaining the ratio in the test cross, dihybrid test cross, 1:7:7:1. So if a linkage phenomena present in the genes, it shows more parental characters are obtained in the offsprings. Less number of non-parental characters. Nearly 97% of the parental characters observed in if linkage phenomena is there. 3% of offspring shows non-parental characters. But in the Mendel ratio, we are getting 9:3:3:1. We are getting 9 + 1, 10 parental characters, 3 + 3, 6 non-parental characters. The non-parental characters ratio is more in the Mendel, but linkage phenomena, it says it is wrong. If linkage is present between the many genes on the same chromosome, most of the characters are parental characters. If any chance is there in the crossing over, definitely it leads to 3% or 4% of non-parental characters are obtained in the progeny. Are you followed? Is it clear? Okay. So sometimes the linkage phenomena observed in the autosomes, that is says autosomal linkage. The linkage phenomena observed in the allosomes or sex chromosomes, that is called allosomal linkage or sex-linked linkage. So autosomal linkage, it is a autosome, which is the genes which are located in autosomes only. Allosomes linkage, the genes which are present on the X or Y chromosome. Generally, X chromosome is we can take into consideration. So we already explaining about autosomes and allosomes. So characteristic features of linkage. What is the significance of the linkage? Linkage involves two or more genes which are located in same chromosome in a linear fashion. So that says that, so if gene is there, so this is the chromosome A, B, C, D, E. These are the different type of genes. These genes are arranged in the linear fashion, and we can see linkage phenomena in between A and B, A and C, A and D, like that. So linkage reduces what? Variability. Linkage reduces variability. And we can't expect a different characters in the next generation. Linkage may involve either dominant or recessive alleles, some dominant and some recessive alleles. If it is in coupling phase, the linkage phenomena observed between the dominant genes. If it is in the transposition, the linkage phenomena observed between dominant and recessive alleles. So it may involve either all desirable traits or all undesirable traits, some desirable and some undesirable traits also. It is observed for oligogenic traits as well as polygenic traits. Linkage usually involves those genes which are located close to each other. So this is very important. When the distance between the genes are very, very smaller, linkage is very strong. Our strength of the linkage is more. So the presence of linkage leads to higher frequency of parental types than the recombination. You can understood this point. The presence of linkage leads to higher frequency of parental combinations than the recombinants in the test cross. So if, for example, no crossing over takes place, definitely we got only parental characters. So if crossing over does not occur, the genes located on one chromosome are expected to be inherited together. Thus, the maximum number of linkage groups possible is an organism is equal to haploid chromosome number. This is very, very important in most of the competitive and entrance examination. How many linkage groups are present in the particular species or the genus? In human beings, in the pea plant, in the onion. So that is equal to haploid chromosome number. Haploid chromosome number. Number of linkage groups are present in the particular species. So we can go for one slide. So that is example, we can explain that. It is here. So linkage groups. Linkage group refers to group of genes which are present in one chromosome. In other words, all those genes which of linkage groups is equal to what? This is very important. You can remember this point. Haploid chromosome number of an organism is equal to the linkage group number. Okay. Chromosome constitute one linkage group. The number of linkage groups is limited in each individual. The maximum number of linkage groups is equal to what number? What number?
Haploid chromosome number.
Yes, haploid chromosome number. So that's why just you can go through this. So for example, in corn plant, the diploid number is 20. How many linkage groups are there?
10.
10. In pea plant, how many linkage groups are there?
7.
7. So in this way, you have to remember in Drosophila melanogaster is 4, human beings are 23, and the pea plant is 7. So these are the examples generally asked in the PG entrance examinations. Detection of linkage. How we can detect the linkage? By crossing over. F1 individual cross with recessive parent plant, that is nothing but is called test cross. What is the main significance of the test cross? Why we are using the test cross? if the test cross, the main significance of the test cross, for example, the tall character maybe exist in this genotype or in this genotype. Yes or no? Okay. The tall plant can exist in heterozygous condition or homozygous condition. How can you know that plant, the tall character is represented by heterozygous condition or homozygous condition?
Heterozygous.
To reveal that secrecy by doing test cross. You reveal this secrecy F1 with recessive parent. So the recessive parent plant crossed with F1 individual, that is known as test cross. By the test cross, we can say that plant is in heterozygous condition. When you are obtained the ratio is 1:1 condition, or 1:1:1 if it is dihybrid. In dihybrid, all four combinations are equally produced in equal proportions are produced in the F2 generation. That indicates our tall character in which condition? Which condition? Heterozygous. That indicates what? That to test the dominant character exists in heterozygous condition or homozygous condition, you can use test cross. Okay, are you followed? Are you followed that point? Test cross is useful to the dominant character can exist in heterozygous condition or homozygous condition. In the test cross result, we are getting 1:1 ratio that indicates our dominant character is exist in which condition? Heterozygous condition. Heterozygous condition. So that's why the significance of the test cross like that. So, and back cross is nothing but the F1 individual cross with any one of the parent, If F1 individual cross with dominant parent plant, homozygous dominant parent plant, that indicates dominant back cross. If F1 plant crossed with recessive parental plant, that is but a recessive back cross. In another words, we can say it is test cross. In another word, we can call it as test cross. F1 individual cross with the dominant homozygous parent plant, that indicates back cross normally. But any one of the parent, I am saying that any one of the parent, it is nothing but a recessive back cross. This is dominant back cross. Recessive back cross is nothing but test cross only. Recessive back cross is nothing but what? Test cross only. In another word, we can say like that. So that is the significance of the test cross and back cross also. So just you can go through that one. So I will go for last slide. A test cross is a breeding or mating between an individual of dominant phenotype who could be either homozygous dominant or heterozygous with an individual that must be homozygous recessive. We know that significance of linkage. So linkage limit is variable among the individuals. Linkage between two or more loci or locus controlling different desirable characters. Advantages for a plant breeder. A linkage between genes controlling two different desirable characters will help simultaneous improvement of both characters. Linkage is undesirable when desirable and undesirable genes are linked together. So the estimates of genetic variance for qualitative characters are greatly influenced by the presence of linkage. So linkage is known very well. The T.H. Morgan and Alfred Sturtevant, who produced the first Drosophila genetic map in 1930. So the distance between the two genes measured in the form of map units. The distance between the two genes measured in which units? Centimorgan. What is that? Centimorgan. 1 centimorgan is equal to 100 morgans. So the distance between the two genes, which is measured in centimorgan. For example, AB, the crossing over percentage is 10%. The crossing over percent, how much it in between the A and B? 10%. This 10% is nothing but 10 centimorgan. The distance between the two genes measured in a map units. The generally unit of map units are centimorgan or morgan. So 10% of crossing over is a chance in between the two genes. That distance become 10 centimorgan. 15% is there, 15 centimorgan. So the percentage of crossing over in terms of distance of the genes only. So this is, you can remember one point also. So first time genetical maps or chromosomal maps or crossing over maps are designed by Alfred Sturtevant. So which is useful to know the position of the gene, know the distance between the genes, know the order of the genes on the chromosome. Why do we construct the chromosomal maps? The use of the chromosomal maps to know the order of the genes, to know the order of the genes. After A, which gene is present on the chromosome? After C, which gene is present on the chromosome? Where is the centromere? And the remaining genes are present on the chromosome. To know the order of genes, to know the distance between the genes, to know the linkage strength in between the genes, we can design what are the maps? Chromosomal maps or genetical maps, or we can call it as crossing over maps, which are designed by first time Alfred Sturtevant along with the T.H. Morgan. In case of Drosophila genetical map constructed in 1913, Morgan was the founder of Drosophila genetics, and in his honor, a recombination map in it is called a centimorgan. 1 centimorgan is equal to 100 morgans. You can remember this point. Are you followed? Yes? Okay. Everybody knows that linkage phenomena? Hello? Hello? Everybody understood the phenomena of linkage? Hello? Are you followed that linkage phenomena? What? Are you followed the linkage phenomena? What is meant by linkage? What is meant by crossing over? Why we are getting different ratio? Just remember that 7:1:1:7 indicates the linkage phenomena in Lathyrus odoratus. Okay. And what is the cis arrangement, trans arrangement? What is the significance of linkage? If linkage is complete linkage is there, there is no non-parental characters. Partial linkage is there, definitely we can see a less number of non-parental characters in the progeny. Is it clear? Hello?
Yes. Yes, anybody else? My dear friends?
If you have any doubts regarding that, please ask me.
Okay, really, sir. Linkage, one of the most important area in genetics. So linkage and crossing over both are most important areas because of the linkage and the crossing over. Both of they are vice versa. So today's class is completely related about the linkage and significance of the linkage. And sir has clearly explained about the Lathyrus odoratus, the linkage phenomena is clearly explained. So my dear friends, regarding of this class, if you have any doubt, please ask our respected resource person. Okay? Okay, sir. Thank you. So tomorrow we will meet same time, sir. Thank you.
Okay, sir. Thank you.